question is Professor Sophien Kamoun of the Sainsbury Laboratory.
There you go. All right. Thank you so much. Huge pleasure, and thanks to the organizers, thanks to Antonis, thanks everyone for having me here. Now we're going to switch to plants, plant disease. So I'm going to tell you that plants get sick. I think everybody knows that, right? If you've been gardening or if you've been exposed to farming, you know that plants get sick. And they really get sick because if you average out a lot of different estimates, but roughly about 25% of our crops are lost to disease, to infectious disease. And that's enough to feed 1.5 billion people. So we do lose a lot to pathogens, all kinds of critters, anywhere from microbes, viruses, nematodes, aphids, what have you. We lose that to crops. We lose our crops to these pathogens. What perhaps you don't know, and we'll talk about that a little later, is that plants have an immune system, and it's a heck of a good immune system. So I'll tell you more about that. Surprisingly, not many people know that, but that's actually a topic and a research topic that's completely embedded in evolutionary biology. And you can't really study an immune system without considering the evolutionary perspective. But before we delve into that, I decided to add this slide because I was sort of inspired by what I heard yesterday. And also Sean Carroll, he is one of my evolutionary biologist heroes. So I had to bring this up here. He has this amazing, amazing book that I make mandatory reading for everyone in my lab. I work with a lot of molecular biologists who are not exposed to evolutionary biology. I think that's a topic, recurrent topic, so I make them read that book. And he has this thing about how DNA is the forensic evidence for evolution. Essentially, if this was in court, it'd be like, without reasonable doubt. And I feel like we haven't heard much about DNA yesterday. I feel we haven't heard nearly enough about it. So I thought I'd start with this slide. For those of you who are not biologists, and really, biology has experienced a huge transformation with genomics. Genomics, what is genomics? Genomics essentially allow us to read the book of life. Really, the book of life is written in the genome, in the DNA. So essentially, before genomics, biologists were studying books without really opening them and reading them. That's a very simple metaphor that I hope you can sort of take with you. So what genomics allowed us is really to read the genome, read the text of that book, open it up and read it. And these days, not only can we read it, we can actually interpret it, we can rewrite it, and we can even create completely new paragraphs of these books. Synthetic biology is an incredible science. So what we're experiencing in biology now, this is very early days of that. Really, fasten your seatbelts, because there's a lot of stuff happening here. And this is, in a way, an extension of evolution, right? What we're experiencing is biology is evolving from a descriptive field to essentially a design-driven discipline. Because now we can create actually completely new genes, new proteins from scratch, using AI and tools like that. So fasten your seatbelts again, because evolution has actually took us to this stage, where not only can we read, rewrite, interpret the genome, but we can even completely design it from scratch. The other thing I want to emphasize is genomics. For me, it was an amazing, amazing thing in my career, because I was an undergrad in Paris. I came to biology as a natural history buff. I grew up collecting beetles and all that stuff. I went to university in Paris, and zoology was taught like in the 19th century. They were asking us to draw stuff, and I just was completely switched off by that. I felt very much more modern. Computers were invented, etc. DNA was invented. I was much more excited about harder science. So I went into molecular biology and molecular genetics, but I kept that interest in evolutionary biology. And then what happens, 15 years later, genomics was invented. And then all of a sudden, I realized that genomics is not just the science of DNA, it's a science of evolution too. Because genomics is about comparing stuff. You can't do genomics without doing any comparative analysis. And really, comparative analysis is what evolution is. Okay, so again, the genome carries the forensic evidence, the forensic record of how life evolved. Apologies, Sean, I like to borrow your statement here. And I just want to add something before I move on to plant diseases, I think it's such like a case, the case so, without, without any doubt, right? If this was in court, like Sean wrote, there'd be no doubt that evolution is a fact given the genome. And I really wish that religions would leave us alone and leave our science alone and focus on the real issues, which are like the moral humanitarian issues that we know we're facing right now in the world. And I'm come from North Africa, so I'm very much sensitive to what's going on right now across the world. Okay, so let's move on to plant disease and plant biology. I have a saying that I really love. My colleagues and I like to say that. Plants save your life every day. Medicine, maybe one day, save your life. Hopefully one day. We heard some really great stuff. Antibiotics, amazing. But plants, really, they save your life every day. Without plants, there'd be no food. And so the UN has actually declared 2020 as International Year of Plant Health. And that's my field. I'm a plant pathologist. I study plant diseases and plant immunity. And plant health is actually one of the major issues in agriculture, in producing crops in a sustainable way. And this shows you that there was a massive increase in a number of diseases and new epidemics. And this actually slide is about only fungi. And it stops at about 2010. And the last 15 years, we had new pandemics that were not even covered by this slide. And the reasons are many, increased trade, climate change, and so on. But pathogens also evolve and jump from one host to another. We heard about the flu virus moving from pigs to humans. This happens also with plant diseases. We get new diseases all the time, taking advantage of those delicious crops we plant in monocultures. And the problem with agriculture is really massive. There are solutions, chemical solutions very often. Unfortunately, genetics is more complicated because of societal sort of resistance to some of those solutions. And this is an example to show you how difficult it is sometimes to bring these solutions to parts of the world. This is a slide I borrowed from a colleague from Punjab Agricultural University. This is the potato blight. This is the Irish potato famine, one of the two major diseases I studied in my career. This field is completely destroyed by the potato blight, and the poor guy is spraying with the wrong nozzle. That's a herbicide-type nozzle. They probably sold him Roundup. That's what my colleague said, which is obviously killing even further the potatoes. He's not wearing any protection, as you can see. A similar farmer in the West would be totally covered with all kind of protection. A lot of issues, his bare feet and everything. And when you have these diseases, this happened 10 years ago. There was a new strain, new race of the potato blight pathogen that moved from Europe through trade to South India, and then a few years later moved up to West Bengal. Massive losses. Many farmers committed suicide. That's how bad it was. They just totally lost their farms. They had to take their farms away from them. That was a disaster. The interesting bit is we have this. We have immunity. What you see on this slide is 3 different types of potatoes. The one in the middle, I'm not going to point, but the one in the middle is fully resistant, as you can see, compared to the two on either side, completely destroyed by the potato blight pathogen. The reason why that plant is doing just great is because of a single gene. That gene codes for an immune receptor. We know exactly how this works now. That immune receptor is enough to make the potato plant fully immune to the pathogen, as if there was no pathogen, unlike the 2 plants next to it, which only lack one single gene. So why is this a problem? And this technology is amazing because every farmer in the world knows how to handle seeds, right? So if you give them seeds with the right genes, they'll be fine. They don't need to train them about protection and fungicides and all of that. The reason why this is not the end of the problem is evolution. So my field is all about evolutionary biology. And just as we heard from Paul, and thank you, Paul, for leading up to this point, the pathogens are amazing, not because they can cause destructive diseases, because they keep adapting at whatever we throw at them. So if we throw at them resistant plants, they'll very quickly, in fact, in our systems, sometimes within one growing season, they make a new race that will overcome the resistance. So let's talk a bit about this topic, and I'm going to take you a little bit to some of the concepts that are underpinning this knowledge. So first of all, I'm going to show you this slide because I think one of the beauty of our system of plant pathogen interactions and as an experimental evolutionary biology system is we can study this coevolution at multiple time scales. We can study this at the Grand Canyon scale, if you like, at tens or hundreds of millions of years, or we can study this as a sand dune ripple scale, which is basically what happens in a farmer's field in 1 or 2 seasons. And what I will do now is I'm going to give you some vignettes of some of the concepts we have in this field, both involving the kind of macro view, if you like, the tens or hundreds of millions of years, and then switch to the more recent stuff that happens even, again, within just a few growing seasons. So first of all, let me introduce some pathogen, which is sort of the connection I have with Antonis. We both work on what we like to call filamentous pathogens. So these filamentous pathogens are, if you see them on a petri dish, or if you sort of get exposed to them, you call them fungi. But actually, this is a term we have to group 2 different types of organisms that are separated by over 1 billion years of evolution, but superficially look very similar. So these are the fungi and the oomycetes. So the oomycetes, maybe a term that's new to some of you, is actually the fungus-like organism that include the potato blight, the Irish potato famine pathogen. These are fungus-like organisms that are actually very deep root in evolution, more related to kelp, for example, but totally unrelated to fungi, but evolve essentially to superficially look like fungi and really have many similarities in their lifestyle, in their life cycle that are similar to fungi. One of the common things that these pathogens have is this capacity to produce incredibly large populations. What you see on this slide is a rust fungus. This is a wheat field in Australia that's infected with the rust fungus. The tractor is plowing over it. And gosh, look at the population size. Look at how many spores are being produced in this one single field. And so the solution that these pathogens came up with is in that particular field, there's probably enough diversity to make new races of these pathogens that will infect whatever plant you throw at them. And the same, if you walk through those fields, some of you may have done that, you'll be covered, you turn orange. Better kind of orange maybe, or I don't know. Somehow when I think of orange, I start like shaking or something, but anyways. Okay, we talked a lot about evolutionary biology and how it's poorly actually sort of medical school and all of that, but it's the same with molecular biology. Really early in my career, we had to work hard to get molecular biologists to understand evolution. One of the things we kept sort of harping about is don't call Phytophthora, the potato killer, don't call it a fungus. So we made T-shirts. Bats are not birds, dolphins are not fish, or mycetes are not fungi, and we kept sort of distributing these. I have to say we moved beyond this. I think many people now appreciate the difference. One of the amazing things about these pathogens is how deep these lineages are. When you look sort of deep in the phylogenetic record, they're really, really deep. They go hundreds of millions of years. And my colleague, Christine Strulu, wonderful paleontologist, she has fossils. She described an oomycete infecting plants from 300 million years ago. 300 million years ago, it's an oomycete. You could recognize the oomycete spores. They're very typical. They're called oomycete because they have egg cells that are incredibly distinct. And you could see them inside the plant tissue. So they were parasitic 300 million years ago. And the crazy things, many of these lineages are obligate parasites. They cannot do anything else except infect the plant. They cannot reproduce unless that spore lands on a leaf that it can infect. So now think about this. Think about this sort of crazy system. These guys are like gamblers. They throw away their spores. They have no idea where those spores will land. And in every cycle, every time they produce those spores, there has to be at least one spore that lands on one leaf that it can infect. And remember, plants have an amazing immune system. So their solution, and actually this was reviewed recently by Antonis in this topic, is in the genome. You look at their genomes, and I think some of you have read in your textbooks or maybe had it in your undergrad class that parasites evolve small genomes. They evolve reduced genomes, right? They lose genes because they depend on the host, blah, blah, blah. Here we have the exact opposite. What we see is these parasites have evolved massively big genomes that are 10 to 50 times larger than the closest non-parasitic relatives. In fact, many of these pathogens have genomes that are larger than the host, larger than the plant genome. So when you look at that interaction, plant, microbe, which one is more complex? Well, you could ask yourself a question there. And what's remarkable, you can see it with these blue, sort of blue balls there, the larger the one, the bigger, is that there are 3 completely independent lineages, oomycetes and 2 types of fungi, that evolved these massively big genomes independently. Convergent evolution, again, very obvious. And so again, this contrasts with the textbook dogma. So I'm going to show you a slide that's a little bit technical, but bear with me. And this is essentially a concept we observed already a while ago, that the big genomes are actually populated with repetitive DNA, transposons. They're infected with transposons. But the genes that matter for the infection, the virulence genes, they're actually located in these regions. So what you see here with the red dots are genes that were experimentally validated as being involved in virulence. And what that means is that they are very unique in their position. They're in the middle of hundreds of kilobases of repetitive DNA. And sometimes you have cases where there's 1 or 2 genes in just 200, 300 kilobases of DNA. That's the only genes that's there. So it's very, very clear signature. We call this a 2-speed genome. Multi-speed genome is probably better, but in any case, the point is that the genome architecture is very unique. And what's remarkable is convergent evolution. This is the potato blight. On the left side is a fungus. It's a plant pathogenic fungus that's separated by over 1 billion years of evolution, evolved exactly the same genome architecture. And what we think is this architecture allows them to be uniquely diverse. They mess up their virulence genes all the time. That population of spores is very diverse. We don't have the technology to sequence all the spores yet, but that will happen. And then we'll find out that a lot of diversity is in those fields to the point that they will be able to find the leaf they can infect. If you think about it, they produce a very uniform population of spores. Very soon it will be game over, they'll go extinct, right? So selection has eliminated everything that doesn't work, and this is the formula that works over millions of years. They can do that gambling, and they will still persist in the biota. Okay, now I'm going to switch tacks to talk a bit about more mechanistic work. And I'm going to tell you a little bit how these guys infect plants. And the way they infect plants is through virulence proteins. We call them effectors in our jargon. Basically, these pathogens secrete all kinds of proteins. Think about them as keys. They have basically key ring full of keys. And they find locks, they find targets in the host, and they open up those locks and suppress plant immunity, for instance, and stuff like that. The interesting bit here is the same keys that they use to unlock plants is also the trigger for immunity. So plants haven't been sitting there being infected. They actually came up with a solution. They came up with immune receptors, sensors, that allow them to basically detect these effectors and activate immunity by detecting the effectors. So now you can see how this is an incredibly unstable evolutionary system, right? Because this pathogen produces these spores. One of these effectors are actually required for them to infect the plant. So the effectors are good when they land on the leaf. That effector is evolving to be good at working on that plant. But if the plant has one receptor, that's enough to turn that effector into liability. And now the pathogen is actually triggering the immunity through that same effector that is actually evolving to be good for that. So this is like why I hate it when people talk about selective forces, because for me in this system, selection is completely fluctuating like crazy. Depending on which spore lands on which leaf, you have a completely different opposite impact on the single gene, single trait in the pathogen, whether it's good or bad. And this is really sort of a plus-minus kind of situation. Either they infect or it's game over. So a little bit, some examples about this evolution. So we have a lot of evolution towards making versions of these effectors that are not detected by the receptor, but still do the business. They still can unlock the key. So we call these stealthy effectors. And I'm going to show you a case here. I'm getting a little bit technical here, so bear with me, but I think it's worth it. So even for the non-biologists here. This is a case from a disease called the rice blast disease. It's caused by a fungus called the rice blast fungus. Actually, the most important plant disease on the planet, because we lose so much food to the blast. Rice, is so important. It's probably enough to feed like over 500 million people what we lose to this disease. And this is a single gene in that particular, single effector in that particular fungus. There's 4 versions of it. One is detected by an immune receptor from rice called PIC, which has been introgressed and bred into rice for thousands of years. And then the other three versions are actually stealthy. They're not detected by this receptor. And the way the receptors detect the effector is through that little orange box there called HMA, and that's a domain that the effector evolved to bind that the receptor evolved to bind the effector and detect it. And so this is what we can do with genomics. This is what we can do. We don't have fossils, but we can basically do the same thing because we can reconstruct all the evolutionary steps that happened. So we know what the ancestral version of the effector is. It's this ancestral version, the one that's detected by the receptor. And we can trace back all the steps under which certain amino acids, there's only 4 amino acids that are different. You see that? There's only 4 letters that are different in this particular effector between the different versions. And we can trace back every single step. And using these ancestral reconstruction type of methods, Joe Thornton in Chicago is the one who pioneered that. Again, another one of my evolutionary biology heroes. But we can basically, we don't have fossils, but we can find essentially all these intermediates through a detective work of molecular evolution. The beauty of this, and I don't know what happened here, but okay, the slide got messed up, but the beauty of this is all those 4 amino acids, all of them are in the binding interface between the effector and the receptor. So all those 4 changes are having an impact. We measure that. We measure binding affinities and all that stuff, right? So, and what's amazing is all the changes in this gene, and for the experts among you, no synonymous substitutions. Only 4 polymorphism, only 4 letters changed. All of them changed the amino acid, all of them in the binding interface. So it sounds a lot like Paul Turner's actually virus, where you have these proteins that are really important for the infection. And then when you have super rapid evolution, these changes happen very quickly in a rice field to the point that you don't even see, you don't even have time for silent mutations to accumulate. Okay, a bit more technical. This is so similar to what Paul showed you a moment earlier for the influenza virus, but this is a rice blast fungus protein. And this is showing you a lot of different versions of these proteins on different lineages of these pathogens. Some of them infect wheat, for example. There's a pandemic actually going on right now with a disease called wheat blast. And you can see that all these polymorphisms are on the surface. And this leads me to something else I find absolutely beautiful in talking about the forensic record. This is a concept called homoplasy. I know Antonio studied this with Sean. And this is an example of conversions, right? If you look at the one on the right there, number 78, right? So that's an amino acid called methionine. Bear with me, it's worth it, really. It's ATG is the letter. ATG is the codon that basically translates into methionine. In this effector, twice, there was 2 times an evolution from ATG, methionine, to leucine. And it's a different sequence. One went from ATG to TTG, and the other one completely independently evolved from ATG to CTG. So we know these are independent mutations, and we know also that they result in exactly the same change, same amino acid. The beauty of this is that's the, okay, I don't have a pointer, but you see M78L, that's actually the residue, and we showed that this is important for the function of both of these proteins. Isn't that amazing? So basically, this is the only way you can make this protein better is by making that one single change. And evolution has done it twice in the fungus. And this happens all the time in the viruses that, by the way, Paul told you. So really, it's no surprise at all. This happened in spike protein of COVID many, many times. Same mutation arising over and over again. What's really cool about this is we actually went back and did the sort of, we call them rewinding the tape experiments. We went back and went to the ancestral version, the ATG, and mutated all the different possible outcomes, right? So not just leucine, but all the other outcomes. And only leucine worked. Isn't that beautiful? And that's why we have conversions. Because that's the only solution that works. So that's the level of detail we have these days. We have these idiots telling us intelligent design, all of that. Anyways, sorry. So I have a few minutes left. Okay. Quickly, I'm going to switch gears and tell you a little bit about the immune receptors. I have to talk about the plant side, right? And actually, this is crazy. So this is really crazy stuff. What happened in my field in just the last few years, and I'm actually heading out on Tuesday to Memphis to give a talk at St. Jude, First time ever I will talk at a hospital, because now we realize that these immune receptors are the same across humans, plants, fungi, bacteria, etc. And that's thanks to structural biology. So that's what you see there. And what's really cool, talking about the evolution of immunity, was inspired yesterday by the fact that this was used in trial and all of that. So these are the same receptors, right? These are the NLR receptors that are also involved in autoimmune diseases. Crohn's disease is a mutation in an NLR receptor. It's the same one that gives that resistance to the blast fungus I was telling you and the potato blight and all of that. The same, basically biochemically, these are the same immune system. In humans, if you go back to 90 million years, which is basically human-mouse split, there's no difference, really, literally. They're pretty much the same receptors. We have an adaptive immune system. That's how we keep up with COVID and all of that. This system is called the innate immune system. It hasn't changed much. And the reason is because the human, mouse, the mammalian NLR receptors, this is a good, by the way, exam question. What they do is they detect conserved patterns on the microbes, like they detect the bacterial flagella, right? So that hasn't changed much in 90 million years, so the receptors don't have to change. In plants, on the other hand, 90 million is tomato and coffee. And what we have in tomato coffee is a completely different immune system, completely different set of repertoire. And during those 90 million years, the receptors have evolved to detect insects, nematodes, bacteria, viruses, potato blight, fungus, everything you can imagine has evolved. And that's because they detect effectors, and these effectors are very rapidly evolving. So the message here, and this message I think that Marlene would agree with, is that the pathogens, the parasites, are actually shaping our genomes. They're shaping the genomes of the host. They're shaping the human genome, but they're also shaping the genomes of these plants in a massive way. Okay, really, literally, I'm going to finish with a very quick thing. And this is even crazier than what I just told you, okay? So I have to finish with this. So something happened in evolution of these receptors that's absolutely insane. So I told you how these effectors bind targets, right? But throughout evolution, some of these receptors, they turned the table on the pathogen. They integrated the effector target into the receptor and turned it into a bait or a decoy, if you like, to detect the receptor. It happened. It's insane. I mean, I only discovered 10 years ago. It's just like when I heard about it, I was shocked. It was a work by French scientist Stella Cesari, beautiful work. And the crazy thing, it hasn't happened only once or twice. It happened like dozens of times. We annotated from 250 plant genomes, 100,000 NLR receptors, and 5% of them have these integrations. So amazing. All this stuff has integrated over and over again, talking about evolution, convergent evolution, all of that. So the question becomes, if evolution has done it, can we actually do it in the lab? Can we just mimic evolution? Can we make now design basically new receptors by flipping this integrated domain by something else? And if you start thinking about it, and this is actually Yorgos Kourelis, a crazy guy in my lab who's half Greek, half Dutch, which is, I think, an explosive combination. He thought, okay, I'm going to put antibodies here. I mean, what if we put human, mammalian, in this case, antibodies? And he did that, and he did the proof of concept, so I'm going to skip it. We call these pico bodies, by the way. It's kind of a fun thing, and these antibodies come from camels. They're not coming from humans. So if he makes like an antibody that detects, for example, a fluorescent protein, the green one, then he can get an immune response against the green protein, but not the red one, and vice versa if he makes an antibody that detects the red one. And they work, they give resistance and all of that. This is a virus that produces proteins. So I can skip all of this. Bottom line, it works. So we can now design new, made-to-order, if you like, receptors that detect pretty much anything by using antibodies and flipping them into these plant receptors. And also now we have AI tools. Nobel Prize last year was given for binders. We can also do this with binders. We don't even have to use antibodies. We can create synthetic proteins that we can introduce in these receptors. So I'll stop there. I think I'm out of time. Thank you for your attention. I really want to always thank the people in the lab. So this is an amazing team. I work with absolutely outstanding people and also outstanding colleagues. So thank you so much.